Welcome! My name is Jana and I will provide you through the comparison of short and long reads in structural and functional annotation of non-model bacteria. The study compares Illumina and Oxford nanopore sequencing technologies. For the comparison, we examined thermophilic producers of polyhydroxyalkanoates, degradable bioplastics. For the reference, we used previously assembled type strain. Particularly, we accomplished structural annotation and quality assessment as well as functional annotation in the form of assigning coding sequences into clusters of orthologist groups. Bacteria obtained from 3 different collections of microorganisms were sequenced using the Illumina HiTSeq and Oxford nanopore MinION technologies. The long reads, as shown in the lower part of the pipeline, were base called using GAPI. Subsequently, genomes were assembled with flying and polishing was done using Kraken and Medaka tools. The processing of short reads is indicated at the pipeline stop. Namely, the short reads were initially adapter and quality trimmed using Trimomatic. Next, the genomic assembly and polishing were done using spades. The quality assessment of the assemblies was performed using Quast. Genomes were annotated using the NCBI prokaryotic genome annotation pipeline, a standalone software package version. The functional annotation in the form of assigning protein coding genes into clusters of orthologist groups was done using the database EGNOC Mapper and the prodigal prediction method. The structural annotation, as shown in the table, revealed the nanopore approach tends to assemble the long reads in a small number of long contigs. In contrast, Illumina resulted in low N50 and high L50 metrics. In addition, more structural features such as protein coding genes were found in the nanopore assemblies compared to the Illumina assemblies. On the other hand, a comparison of the results with the type strain previously assembled using a hybrid assembly approach revealed nanopore tended to overestimate the number of structural features. Comparison with the reference also revealed the nanopore assemblies to be error-prone and contain more mismatches and indels than the Illumina assemblies, even though the basis accuracies were improving by polishing. In this slide are shown results of the functional annotation in the form of the number of protein coding genes assigned to each cluster of orthologist groups for each bacterium and sequencing technology. The type strain, used as a reference, confirms that nanopore assemblies tend to overestimate the number of features. The difference in the number of genes assigned to each group between the reference assembly and the Illumina assemblies is on average 33%, but for nanopore the value reached 53%. The overestimation was evident across all COGs. In conclusion, nanopore assemblies have proven to be a more efficient approach for initial insight into the genome. Namely, the Oxford nanopore technology resulted in a small number of long contigs. Moreover, nanopore assemblies revealed more genomic features in comparison to Illumina assemblies. In contrast, the Illumina approach is more applicable for the detailed study of specific genomic regions. Illumina approximates the number of individual features and contains fewer errors than ONT compared to the reference genome. This brings us to the end of the presentation. Thank you for coming to overview of our work. If you have any questions, Do not hesitate to contact me by email or during the conference. You are kindly welcome to view my resume via the displayed QR code.